the University of Calgary. We're going to start with our University of Calgary one, but yeah, for future seminars, we'll have one for Alberta. So um, APRN and some of our trainee members are part of the University of Calgary, which is located in the heart of southern Alberta. And we both acknowledge and pay tribute to the traditional territories of the peoples of Treaty 7, which include the Blackfoot Confederacy, comprised of the Siksika, Piikani, and Kainai First Nations, the Tsuut'ina First Nations, and the Stoney Nakoda, including the Chiniki, Bearspaw, and Goodstoney First Nations. The state of Calgary is also home to the Métis Nation of Alberta, Districts 5 and 6. As we are here to discuss pain and medicine, I'd also like to acknowledge that Indigenous people are disproportionately affected by chronic pain and also the contributions of Indigenous people and medicine in our daily lives. Predating what is referred to today as medicine, Indigenous people have been creating treatments and cures using plants and animals for thousands of years. Indigenous people brought forward several natural treatment options for different sicknesses, many of which were adapted by Europeans. This includes the active ingredient in aspirin, and aspirin is widely used to manage acute pain. And the science of salicylin in modern medicine in aspirin is an example of the importance of traditional Indigenous plant knowledge in human health and modern science. And we wanted to thank everyone for joining us today as part of our third APRN seminar of 2025. And thank you to our speakers and everyone in attendance, patients, scientists, and clinicians. We'd like to start off this presentation with an emphasis on intentionally creating a brave space where everyone can feel safe to ask their questions, engage in active listening, open and honest dialogue while respecting diversity and diverse perspectives. We will be recording the speakers' presentations, but we will turn off the recording for the question period so that people can feel more comfortable asking any questions they have. With that, we're going to start off with our first speaker, Dr. Zochodne, who's going to be introduced by Margaret.
Yeah. Dr. Douglas Zochodne, he's a neurologist and neuroscientist at the University of Alberta, where he is the Divisional Director of Neurology and Director of the Neuroscience and Mental Health Institute. He was trained at the University of Western Ontario and Mayo Clinic, and his career has focused on peripheral nervous system biology and diseases with faculty positions at Queen's University, the University of Calgary, and the University of Alberta. His past roles including editor-in-chief of the Canadian Journal of Neurological Sciences and president of the Peripheral Nerve Society. He's a fellow of the Canadian Academy of Health Sciences. He investigates diabetic polyneuropathy and peripheral neuron regeneration, with recent work highlighting intrinsic molecular mechanism in an adult sensory neuron plasticity, including repurposed tumor suppressor pathways. Continuously funded since 1988, his lab has published over 280 papers and chapters and currently focuses on molecular mechanisms of peripheral neuron plasticity, regeneration, as well as diabetic resistance. So without further ado, thank you so much for being here, Dr. Zochodne.
Well, thank you very much for the introduction, and pleased to join APRN. I should just add an update that Dr. Penny Smythe is now our division director in neurology, but I was pleased to serve that in that role for a number of years. I'm going to try to move quickly so we have enough time to set up Dr. Shiers, who has some exciting new work, and hopefully this will paint a bit of background too from what I know of their new directions, which are quite exciting. Here's our University of Alberta Treaty Acknowledgement. We're located on Treaty 6 territory, a traditional gathering place for diverse Indigenous people, including the Cree, Blackfoot, Métis, Nakota Sioux, Iroquois, Dene, Ojibwe, Saulteaux, Anishinaabe, Inuit, and many others whose histories, languages, and cultures continue to influence our vibrant community. And to just add what was just mentioned, the impact of diabetes mellitus itself on First Nations communities, both in the United States and worldwide, has been marked and challenging. Right. So this is about peripheral neurobiology. Hope to convince you that the peripheral nerves are the house of their own types of disorders and pathology, and you have to understand them differently than the central nervous system. So it's not really a microcosm of what happens in the CNS, although the CNS challenges for regeneration are yet greater. And so this is a peripheral nerve trunk. The nerves ramify through the entire body, permeating all organs except the interior of the brain and liver. And similarly, diabetic neuropathy is a widespread disorder involving peripheral nerves. And you see in these fascicles of unmyelinated and myelinated fibers, both are targeted by the disorder. Um, and there's a unique and compelling beauty to it. So these are individual fascicles, but each axon is intimately connected with the Schwann cell, which is a major determinant of axon health and viability and conduction. But we're particularly focused today, and I think Dr. Shiers will pick up on this, is on sensory ganglion, where the cell bodies of the sensory neurons exist. Well, diabetes targets motor, autonomic, and sensory fibers, as well as the CNS. Um, the sensory involvement can be early, predominant, and associated with neuropathic pain. Um, Right. So at the other end, however, of the sensory neuron is the skin innervation, which is an area that we've spent a lot of focus on, where the fine epidermal fibers ramify through the skin here. Um, Sarah, can you see my pointer? Okay.
We can see it.
Okay, great. Thanks.
We can
And
see
here
it
you
now.
can see in immunohistochemistry these fine terminals that are continually growing. So our argument has been that in diabetic polyneuropathy, the sort of growth potential or the plasticity potential to support distal fibers in the skin like this is impaired. Um, so I don't have to remind this group that the prevalence of diabetes mellitus continues to explode. Some severe hotspots. I mentioned indigenous people in Canada, but certainly the Middle East and Central America and South America as well. Um, but also, it's not a late complication of diabetes. Children can develop diabetic neuropathy. And here's some older work by Wayne Rundles from Ann Arbor showing that children at ages 17 to 20 can have symptoms of polyneuropathy. So it's not a late stage complication. And I'll also argue that it's not a microvascular complication, despite that terminology for many years. So just the overall classification, distal symmetrical polyneuropathy is by far the most common complication, and it's a diffuse disorder. Symmetrical can involve large myelinated fibers or small fibers as well, and may have a component of autonomic involvement and occasionally motor involvement as well. And I'm not going to mention much about focal neuropathies, except to say that not only does diabetes target these sensory axons, this is so-called stocking and glove sensory loss in the arms and legs. This could be pain, this could be tingling and paresthesia as positive sensory symptoms, or it could be numbness or loss. But it also impairs the regenerative capability of sensory neurons as well. So if there's a superimposed injury, there can be delayed regrowth, such as carpal tunnel syndrome from a focal neuropathy. But also, if one were to end the hyperglycemia of diabetes and allow the nerves to recover, there's a regenerative deficit there that is of interest. This is just dry skin and an insensate foot. And of course, the worry is the risk for amputation. So here are some original descriptions of positive sensory symptoms, such as numbness, tingling, paresthesia, shooting pain. I was told in Rochester, Minnesota, they use the term constriction rather than tingling, um, cramps, aches, and pains, burning pain, difficulty sleeping, opioids not very helpful in general. And the risks, of course, being poor healing of injuries, self-injuries, and of ulcers, and risks for amputation. And here's a so-called Charcot foot. It's an insensate joint that can have accelerated osteoarthritic breakdown from lack of sensation from neuropathy. So it's just some key points here. 50% of diabetics. And you can imagine with the prevalence ballooning worldwide, how many people have evidence of polyneuropathy. Symptomatic neuropathy, though, maybe in about 20% of diabetics. I mentioned the positive sensory symptoms, um, stocking and glove distribution. The longest fibers are targeted first. That's why the tips of the toes and the fingers are the site of the earliest symptoms. Autonomic involvement may accompany, motor involvement usually later. And there may be these additional focal neuropathies that I'm not going to talk a lot about. Major risk factor for lower limb ulceration. And I think this says all you may want to learn about initially from neuropathic pain from diabetes. This is from an old drug advert, but the patient has so-called allodynia. They have burning at night. They don't want the bed covers to touch their feet because of its induction of pain. Uh, so there are other causes of pain in diabetes, though. You have to think about other causes. Neuropathic pain, I think, should be strictly limited to neuropathy pain, although people use it sometimes to describe CNS pain. The descriptors I've already talked about. 7 to 20% of diabetic subjects. I mentioned allodynia, in which innocuous sensory stimulus generates pain, and it can be cold, touch, or pinprick. Um, and some persons with diabetes mellitus may have prominent pain, but without a lot of actual damage. And our treatments are inadequate, but there are some treatments out there. Right. So how do we approach this disorder and how do we attempt to reverse it? And I've already started to make the argument that I think learning about the sensory neuron, why its plasticity is impaired, why these terminals can't regrow back into the skin. And I think there's something closely linked between that failure, the sensory neuron, and the induction of pain, whether it's abnormal disposition of the ion channels. And you may hear more from Dr. Shiers about this. It's been a long history of failures to reverse this disorder, which I would call a neurodegenerative disorder, prominent sensory neurodegenerative disorder. We tried a variety of aldose reductase inhibitors in the 90s. There's still one marketed in Japan, um, epirestat, but it's not a magic bullet. We tried growth factors, which were unhelpful, and antioxidant treatments like acetylcarnitine. And there are others out there that people have tried. Um, and some new ones that are of interest, some local therapies that we've been a part of. A new clinical trial should be coming out with Dr. Brill and group looking at some local therapies to regrow neurons, regrow axons in the skin. But let's just focus again on the dorsal root ganglion here. So it's the site of the cell body of the large and smaller caliber sensory neurons. They're unique structures. They're very difficult to sample. Many autopsies don't collect DRGs. You have to look for them. They're hard enough to sample in animal models, let alone people. And so very, very few studies had been done of what happens to the actual cell bodies of the sensory neuron in diabetes. Back in the 60s, there was one study by Greenbaum et al., but the histology wasn't up to speed, and it was unclear whether there's actually loss of neurons, change in plasticity, etc. Again, I'll just remind you that loss of these fine green epidermal nerve fibers are the hallmark of how the neurodegeneration translates into neuropathy. And I have a little schema, don't mind me, that just kind of illustrates how I picture this. So we have termination of the terminals in the epidermis, and with time they gradually retract, terminal retraction. And we think this happens a long time before there's any frank loss of neurons centrally, if there is any at all. There may be some atrophy here. There's changes in the molecular makeup of the DRG over time. There's axonal atrophy, changes in the neurofilament distribution, and this may progress over time. And maybe a little later on, you get a degree of sensory neuron actual dropout and loss. And that's a tough thing because it's hard to treat or reverse the disorder if the sensory neurons are lost completely. So, and this is the ultimate effect that I've already shown you, the stocking and glove loss. So what we want to do is try and get into a stage about here where we know there's loss of fibers, but the sensory neurons are still preserved, and see if we can reinnervate the skin. And we think there's a few different ways to do that. One is to locally approach it. And I mentioned there's a new clinical trial that's of interest to phase 1, 2 to look at a way to increase the growth of these axons. Um, but another way too is you can manipulate the cell body and instruct it to do better with regeneration programs. Um, get rid of that. Right. So we think it's a progressive stocking and glove sensory loss, and it's the result of combined insults and failure to support the neuron. So much of the literature is focused on what's wrong in the environment. Nitrative stress, oxidative stress, lipemic stress, these polyols that I mentioned that were treated with aldose reductase inhibitors. So lots of information about that. But I think my take is that these are going to be hard to reverse and they're diverse and they're of varying importance despite what you hear, including the ischemia. For example, these ganglion may not be ischemic at all. But supporting the neuron, there's some serious failures that we may be able to address. One obvious one that we were interested in a while ago is that of insulin as a growth factor. And insulin receptors are expressed on sensory neurons. And in culture, if you grow sensory neurons and add graded doses of insulin, you can increase neurite outgrowth with them as well. If you overdo it, though, what happens is the neurons can develop so-called insulin resistance, something you may hear about in type 2 diabetes and refers to difficulties in glycemic control. But neurons also develop resistance to the trophic impact of insulin over time. So it's not the complete answer. We need new solutions. But you can deliver insulin near nerve and small doses or in the ganglia, and you can locally improve indices of neuropathy. This was work a number of years ago we did with Ash Singal. It could improve conduction velocity in diabetics with insulin just locally near the nerve. And then Valentine Brussi was a postdoc in our lab who gave very small doses of intrathecal insulin here as well. So getting right to those root sleeves where the dorsal root ganglia are, delivering insulin right where the receptors are, and compared it to controls where the insulin just simply went subcutaneously. And since the doses were small, this didn't influence the blood sugars at all. They were the same as ever. So we weren't improving neuropathy from reversing blood sugars. But we could improve motor conduction velocity and sensory conduction velocity. And it had similar effects to IGF-1, which cross-reacts with insulin receptors. So here's a kind of a schema here, insulin receptor here, some molecular machinery to translate this into impacts on the neurons. So we have the ligand of insulin, go through the PI3 kinase pathway. Notice P10 here. This is an important inhibitor of the intrinsic growth factor signaling pathway. AGEs are advanced glycosylation end products. They're part of the disease that impacts neurons. So in type 1 diabetes, we're missing the ligand. Type 2 diabetes, there's resistance in this machinery to the impact of insulin and signaling. I'll just throw in here because it's really topical is that extended forward with these GLP-1 inhibitors, in addition to helping with glucose control per se, do have direct effects on neurons as well. And I don't think the clinical trials have shown this yet, but I think they might eventually show that the GLP-1 agonists improve neuropathy. And this was using extended-4. And there's a couple of other labs that showed this around the very same time that we did, both in type 1 and type 2 animal models of diabetes. I think Stephanie's going to get to this with much newer RNA data in humans, but this was in a long-term diabetic mouse model already. I think we're the second ones to do this. Tomlinson's lab looked at this a few years earlier as well, looking for unique transcripts. We also looked for superregulatory control here, and looked at miRNAs, and one interesting one was this LET7I, just as an example, expressed in dorsal root ganglion sensory neurons, increases neurite outgrowth, and also improves diabetic neuropathy in models. And these were a list, and you can't read them, but don't worry about it. These are a list of a variety of miRNAs that are altered in diabetes. Yeah, and that's just the data that showed it improved mechanical sensation, thermal sensitivity. In this model, the higher bar means that there's less sensation. And depending on the model you use, if you look very early after the induction of diabetes, you may see hyperalgesia as a pain model. Later on, there's loss of sensation. I think both are related to the neurodegeneration. First, abnormal signaling, and then loss of sensation, and then the conduction velocities. You do conduction velocities in mouse and rat models. And then, of course, the loss of fibers in the skin corrected by supplementing the animal with this microRNA. So we've had a number of hits, which I won't have time to go over. I mentioned insulin, GLP-1 agonists, near nerve, intrathecal. Some of these hits came from the microarray studies we did a few years ago. CWC is a spliceosome protein, so that's interesting. And then more recently, looking at a number of sort of more general, widespread acting growth molecules that are known about in oncology, but are expressed physiologically in neurons and DRG neurons and may have an impact. And I'll just kind of end here, hopefully on time, with just Wang Fem's paper with the group looking at insulin combined with a PTEN inhibitor. Remember I mentioned that PTEN inhibits the normal insulin growth signaling pathway. And so if you knock down PTEN, we've shown that that improves peripheral nerve regeneration, but also improves regeneration in diabetics and improves neuropathy here as well. And there were some additive effects with insulin together. And I won't go through it all, but they were in behavior, in conduction, without impacting glycemic levels. You may remember that final growth pathway included PAKT, and here we see that with the PTEN knockdown, you can improve DRG expression of PAKT by knocking down PTEN or using insulin. And this is just to show knockdown of PTEN in the model here. You could improve, these are transverse sections of sensory and motor nerves, or in mixed nerves, I should say. You can improve the caliber. There's axonal atrophy that occurs with PTEN and insulin, and it could improve reinnervation of the skin as well. So an interesting pathway, but one of a number I think we're going to have the opportunity to explore as we learn more about the range of molecules that might be abnormal in diabetes and how to manipulate them. I think quite an interesting number of hits came from the early microarray studies, and I'm thinking with Dr. Price and Dr. Shiers, with their access to human studies, that knowledge of potential targets will grow, and I think it'll be quite interesting to see that. Here's the group. I won't go through all their names from U of C when we worked there previously, particularly Chu Cheng, who was a keystone to the lab, Bagat Singh, who worked on diabetes and PTEN, Jose Martinez, and then more recently in Edmonton, hanging out on White Avenue. So I'll stop there and look forward to Dr. Shiers' talk. Thank you. Oh, and just a heads up, if you're interested in regeneration, this will be out in September. Plug for our book. Thanks.
Thank you so much, Dr. Zochodne. What an amazing talk. Let's transition over to Dr. Shiers. Margaret, if you want to introduce.
Yeah, of course. Dr. Shiers is a research scientist at the Center for Advanced Pain Studies and the project lead of the Human Tissue Procurement and Processing Core at the University of Texas at Dallas. She specializes in histological and transcriptomic approaches to study the human peripheral nervous system with the goal of identifying novel therapeutic targets for chronic pain. Her recent work uncovered Nagote nodules, which are pathological structures associated with sensory neuron death, and dorsal root ganglia from organ donors with diabetic peripheral neuropathy. Using multiplex staining and spatial transcriptomics, Dr. Shiers molecularly characterized these nodules, revealing previously unknown ligand receptor interactions that may contribute to pain in diabetic neuropathy. Beyond her scientific contributions, Dr. Shiers is passionate about revealing the inherent beauty of the nervous system through her imaging work and sharing the art of neuroscience with both scientific and public audiences.
Thank you for the introduction. Hi, everyone. I'm Stephanie. I am going to turn on my laser pointer. So, thank you so much for the opportunity to be here and to share my work. I am really excited to share this project. I've been working on this for about 2.5 years. I'm not actually a diabetic neuropathy researcher. I just kind of stumbled upon something while doing experiments over the last 2.5 years that just seems strongly, strongly linked to diabetic neuropathy. Our manuscript was actually just published and went online last Monday. So this is the first time I'm going to be talking about the entire story uh, in its completion since it was published. So I'm really excited to share and hopefully you find it interesting. So I was told this was a mixed audience, so I figured I'd start off with some really simple background and then try to get into the science. So there
Just
is going to
before you
be.
start, we're just seeing the different screen.
Oh.
So just go to display settings and swap the screens.
um, how does that work?
So just the one in the middle that says display settings.
Does this work now?
Yep,
Oh,
there you go. It's
Yeah, okay,
perfect.
cool.
Awesome. Thanks
Sorry
so
about
much.
that. Okay. So I was told this was a mixed audience, so I do have a lot more background heavy basic science stuff at the beginning. So let me get started. So I was told to give a preclinical perspective on my research as a preclinical neuroscientist. This is opposed to clinical research. So when I started the PhD program back at UT Dallas in 2015, I was kind of told something really simple about the difference between clinical and preclinical researchers, which is that clinical researchers study people and preclinical researchers study animals. And this is kind of true, but I might not fit this definition anymore. So clinical researchers are mostly focused on working in the clinic, having more real-world applications, working directly with patients, looking at human health and patient outcomes, and applying a lot of what we find in the preclinical laboratory setting to human health. Preclinical researchers do work with animal models, in part, mostly rodents. We model human disease in rodents to better understand the underlying biology of human disease so we can find cures or treatments. But I'm mostly a preclinical neuroscientist that studies humans now. And in a typical day, you'll actually see me in the laboratory looking through a microscope or at the lab bench, staining slides that have sections of human postmortem tissue. So we're actually using tissues recovered from organ donors to better understand human biology and the presence of human disease. So why are we studying human nervous tissues? Why are we no longer studying rodents? I actually haven't even done an animal research experiment in over 5 years, even though all of my training for over a decade was in animal research. And this was really for a variety of reasons, one of which I think is probably the most important, is summed up nicely in this paper that just came out a couple years ago, where they looked at estimates of probabilities of successful development of pain medications. And they have a statistic in this paper that says that 93% of all analgesics, so all painkillers, designed to treat neuropathic pain have failed clinical trials. So just think about that for a second. 93% of all painkillers that have been vetted preclinically, molecularly, biologically in animal models for, a decade or more and have worked to treat animal pain are not working once they get into the clinic and start being used in humans. And so this has been blamed on a lot of different reasons. We're not really sure what the problem is, but one thing that we thought might be going on is something preclinical, which might be that there are just differences in the underlying biology between rodents and humans, particularly in the pain sensory system, that could explain why these drugs aren't translating from animals into humans. So in early 2020, our lab completely shifted gears from doing animal research to now working with human specimens. And we did this by partnering with the Southwest Transplant Alliance. This is an organ procurement organization in Dallas. They're about 15 to 20 minutes from our campus. They service all of the Dallas Metroplex. And around 2 to 3 times a week, researchers on our team, including myself, are going to local area hospitals and working with these really amazing transplant surgeons down here to procure these nervous tissues from organ donors. We just had our 382nd recovery. So 382 organ donors who have graciously given us their tissue for research. And this operation started in March 2020. So actually, I went to our first recovery, and then 3 days later, we were all in quarantine due to COVID. So we had literally one organ donor's worth of nervous tissue in quarantine. And then I got to come back to the lab with special permissions from the university and do COVID-19 research during the pandemic with that one organ donor's worth of tissue. So it was super valuable, and it's such a privilege to be able to do this work. So the tissue that I'm going to be talking about today are the dorsal root ganglia. And just so you have a little background on this, although you just heard about them, I'm just going to give you a little bit of an anatomical perspective of what these look like. And I'll show you one from a human. You actually have 62 of these, give or take, in your body. 64 if you include the two that are really large and underneath your brain. Those are called the trigeminal ganglia. And these dorsal root ganglia are in pairs along your spine, your spinal cord. So here's the spinal cord. And then, so for instance, your thoracic 4 segment of your spinal cord has a nerve root coming off on both sides. And there's this swelling, this bulb-like shape attached to this nerve. That's where the dorsal root ganglia is. So this is the T4 ganglia on one side, and then the T4 ganglia on the other side. So when we go into the OR, the surgeons actually remove the ventral portion of the bone, the vertebral column, and what you can see underneath is the spinal cord with the dorsal root ganglia coming off. This is what it looks like. So this is dorsal root ganglia from a human, has this like yellow tint to it. It's actually really easy to spot when we're in the OR. And then you can see these nerves coming off. So these sensory ganglia, these dorsal root ganglia, are jam-packed with the cell bodies of sensory neurons. And these sensory neurons have a really unique shape. They're called pseudounipolar. They actually have a cell body that's completely bald, except for one little stem axon that comes off of them that bifurcates. And one branch goes off to the central nervous system, and one branch goes out to innervate your peripheral tissue. And these communicate information about all sensory stimuli that you can detect in your environment. So for instance, if you were to touch a hot stove, you'd be activating sensory axons that are innervating your hand. Those would send an electrical impulse all the way to the spinal cord and ultimately up to the brain, leading to the sensation of pain. And those cell bodies of those sensory neurons are in the dorsal root ganglion. So this is where we are mostly focused on to understand these cells. So in a typical day in the lab, especially when we got so much tissue, we have so much tissue now from all these organ donors. I've been doing just a morphological assessment using a variety of different histological stains to look at dorsal root ganglion anatomy. And one of the stains that I use is this hematotoxin and eosin stain. It's really easy to do. It takes like 20 minutes. And I've just been screening DRGs from these organ donors for the last few years. So in the hematotoxin labels the nuclei, this like dark purple color, and the eosin labels the cytoplasm of cells, this like pinkish color. So you can see lots of neurons everywhere, these larger cell bodies. And then you can see a lot of nuclei everywhere. So these are a mix of immune cells. Mostly they're actually Schwann cells. And then the cells that are ringing the cell bodies of the neurons are the satellite glial cells. So this is typically what I see when I look at a dorsal ganglion from a human organ donor. However, while I was just going through this, screening process, I noticed that there were some dorsal ganglia that looked quite strange. And so hopefully you can see the difference. Let me just zoom in. Um, you can see there's these like clusters of nuclei scattered about this ganglion, and there are a lot of them, and they're outside the neurons. So they're not neurons, obviously. Um, but I knew exactly what these were because of Dr. Zochodne's work. Right when we had started this organ procurement operation back in early 2020, I had led a journal club for one of our lab meetings about some of the pathology that I'd found in the literature, particularly on his paper from 2004 that had shown these dystrophic axonal swellings, but also for the first time introduced me to what a Najat nodule was, or a nest of Najat. Nobody in our lab, including the PI of our lab, had ever heard of them. Most people in the pain field have never heard of them, especially in the preclinical pain field, have never heard of them. And so, what these or what the literature claims these are, is dead sensory neurons. So the cell body of the neuron has decayed, and what's left behind now and occupying that space is a cluster of what people claim are satellite glial cells. So when I started seeing these things popping up in DRGs from humans, I was like, well, I'm going to go learn everything I can about these. So I went to PubMed and I typed in nest of Najat, Najat nodule, all the terms, and came out with only 28 papers, which in the scheme of science is barely anything. Most of these papers were published in human. They're mostly case studies, single case studies of or autopsy reports of pathologists just looking at a variety of tissues and then seeing if different types of pathologies are present, particularly in the DRG. They would look for the Najat nodule. It doesn't necessarily mean they'd find it, but they would still come up in a PubMed search. They had been found in HIV, diabetes, and then a lot of rare diseases, genetic diseases, even West Nile virus. Very few studies in other species. Interestingly, only 2 studies in rat, and no studies that I could find, at least in a PubMed search, came back for being found in mice. And even in the rat studies, these 2 studies, these animals were treated with neurodegenerative drugs, so chemotherapeutic or even mercury poisoned. So this seems to be something that's really prevalent in humans, but maybe not so in our preclinical animal models. So I had stained 90 DRGs at the time from 90 different people. And what I did was just generate a qualitative scoring system where I like ranked high to very low the presence of Najat nodules in these DRGs and scored them, scored all of them. And then what I did was I went through the medical history to see if there was any themes. And lo and behold, what came back was diabetes. So the medical history we get is a summary of hospital records from the Southwest Transplant Alliance, the organ procurement organization. They summarize the hospital records and they summarize information that they get from next to kin. So they're not always the most detailed. Sometimes they are. So I just had to be as transparent as possible, especially when I was writing our manuscript, on how I grouped these organ donors. So we know for sure if they had diabetes because this is something they're monitored for while they're on life support. Um, so we had 48 donors that were non-diabetic. They had very low Najat nodule scores. And then we had our diabetic groupings where we had diabetics that didn't have any evidence in their medical histories that they had neuropathy and they weren't taking any painkillers. Um, they still actually had higher scores and significantly higher scores than the non-diabetics. And then the diabetics that were taking analgesics but didn't actually clarify why they were taking analgesics. But these analgesics would be things like duloxetine or gabapentin, which are widely used for nerve pain, specifically duloxetine, which I think is the FDA approved for diabetic nerve pain. And then there was 9 donors that actually had confirmed neuropathy, and these all had significantly high Najat nodule scores. Interestingly, we also had some organ donors that weren't diabetic but had other types of pain. Um, but it seemed like the Najat nodule scores were most high in donors that had evidence for neuropathy. But we also had several that had arthritis, but they had really low Najat nodule scores. So this seems to be something tied to neuropathy. So since all of the 28 papers that I could find on Najat nodules claimed that these were dead sensory neurons, and clusters now that are of cells that are occupying the space of a dead sensory neuron, and that these are satellite glial cells in particular, I decided to actually stain for satellite glial cells because I actually couldn't find any molecular papers showing that these were these cells. So I stained for peripherin, just a sensory neuron marker, labels the cell body of sensory neurons and their axons, and then I labeled for GFAP, which is a satellite glial cell marker, but also labels non-myelinating Schwann cells. And then I co-stained with DAPI. So this is a human DRG from a diabetic neuropathy donor. And so GFAP clearly labeled satellite glial cells. You can see them ringing the sensory neurons. There's, the classic non-myelinating Schwann cell pattern, but also very intensely labeled the clusters of nuclei, or the cells that form the Najat nodules. Um, this made it really easy to quantify, actually. So what I found was actually around 25, upwards of 50% in this one donor of all of the neurons had this morphology, had a Najat nodule morphology. And so if these are actually dead sensory neurons, that suggests that around 25%, maybe up to 50% in this one donor of all the sensory neurons are dead in DPN or degenerated in DPN. I'm not showing it here, but we also did do some follow-up studies, and we did show that the population of neurons that seems to be dying are the small diameter neurons, which are and those neurons were also SCN10A positive population. So it seems like the nociceptors were the first ones to actually die off. What was more surprising and what I was really excited about was that when I did the peripherin staining during this experiment, and took some really nice confocal high resolution imaging. Intertwined with the cells at Najat nodules, these clusters of nuclei, I found a axonal fiber bundle. This I could not find in any of those 28 papers. I didn't think this had ever been reported on. Here's some other pictures of them. And here's just a side by side so you can see how strikingly different just a peripherin sensory marker stain looks between a diabetic peripheral neuropathy donor and a non-diabetic. So you can see a lot of sensory neurons being stained. You can see some scattered axons in the non-diabetic. And then in the DPN donor, you can see these 3 Najat nodules with all these fibers intertangled with them, intertwined with the cells there. You can see some sensory neurons here that have this like meshwork of axons around them, so like a paracellular nest, which has been implicated before um, in other rodent models actually, but for other types of neuropathies. And then also some dystrophic axonal swellings, which Dr. Zochodne also had described in another paper, which I don't have time to talk about. So I got really excited when I found these axonal bundles because I was like, wow, this is the first time something's anatomically been described. I'm going to get to name something after myself because that's what you do when you find something. So I was like, oh, I'm going to call this the Shiers neuroma or something interesting. But I was very incredibly wrong because since there was no information on why everybody thought these were satellite glial cells, and there was no information on why people thought these were dead neurons, and so on, I ended up finding the original publication that first described these structures, um, published by Jean Najat back in 1922. It's actually uploaded as a Google book by University of California Library. Has a library card in it still, that was last checked out, 1955, which I thought was really cool. So I don't speak French, even though I am French Canadian. I don't speak French. But I use Google Translator and some AI tools to translate the entire text. And lo and behold, he found those axonal arborizations, these neuroma-like structures in Najat nodules. So he called them residual nodules. He didn't name them after himself. It wasn't named after him until much later. Um, but he found these sprouts within these structures. And I'm going to just read a couple of his quotes because I think they're really nice, especially how scientists write back then. It's just really cool. So, as the nerve cell corpse is reabsorbed, the satellite cells proliferate, and when the nerve cell has disappeared, they form a nodule. And here's another drawing of, those axonal sprouts. Extremely rich bouquets of fibers arise from the glomeruli. So the glomeruli are just the stem axon of the sensory neuron before it bifurcates. Of the surviving nerve cells, which will flourish in the neighboring residual nodules, formed by the satellite elements of nerve cells. So this is really interesting. So what he thought was that sensory neurons, and what he observed was that sensory neurons that are still alive actually sprout fibers off of their cell body, off of their glomerulus, and their glomerulus, their stem axon will actually swell as they sprout these fibers, and they will sprout fibers around dead sensory neurons that are actually in the state of decay. So this is a dead neuron that's in the state of decay, and now all these fibers are kind of ensheathing it. And this is really weird because sensory neurons are not supposed to sprout neurites from their glomerulus, and they're not supposed to sprout neurons from their cell body, especially. They're pseudounipolar. This is what gives them their unique electrical properties. They don't, they're kind of bald. They don't have dendrites or, processes coming off of their cell body. So this is a really, really unique finding. So I explored this in a variety of ways. One way was to look at just what kind of sprouts are these axons that are forming Najat nodule arborizations. I found that they're not sympathetic axons, they are sensory axons. They look to appear to be mostly nociceptive axons, so they seem to be sprouting from nociceptors. So they express TRPV1 and now 1.7. And then I did something that was really challenging actually because I'd never done it before, but I took these really thick sections and then I took these high resolution Z stacks at high magnification through the Z plane and used a MARIS to trace the axons um, of the Najat nodule to see where they were actually sprouting from. And turns out that in pink, you can see all the axons that are sprouting off of the cell body of the neuron and its glomerulus. There is some connection to the Najat nodule, but most of the actual connection of the Najat nodule comes from its these dystrophic axonal swellings. So these sensory neuron fibers that are sprouting off the cell body become damaged. They start trying to regenerate. Um, they degenerate, regenerate, and they make these swellings. And those sprouts then go on to form more sprouts that form into the Najat nodule arborizations. So they don't look these neurons don't look pseudounipolar anymore. We also see the same thing recapitulated in neurons in culture. And so I know I'm running short on time, so I'm just going to briefly go through what we did next. But we use spatial transcriptomics, which gives us a snapshot of all the mRNAs that are found in Najat nodules. So you can see these barcodes that are touching Najat nodules. We identified these and then looked at what mRNAs were found there. We did this to answer a number of questions. One of them was what cell types actually comprise Najat nodules. There's this cluster nuclei, and everybody says they're satellite glial cells, but are they? Turns out that the two main estimated sources of mRNA transcripts, two most probable cell types that are there, came back as non-myelinating Schwann cells and satellite glial cells, which we confirmed with some RNA-scope and C2 hybridization. The majority actually do look like non-myelinating Schwann cells with some satellite glial cells in there as well. What was more interesting and probably most important, which we're hoping this will just, this paper was really to establish this biology and bring it to the surface for the preclinical researchers to even be aware of it being present in human, but now to give them some drug targets, mechanisms potentially that they could go after to either protect sensory neurons from dying or to prevent the sprouting from happening, is this data set. So this was all analyzed by this really talented graduate student, Khadija Mazar, where she looked at interactions of ligands, so mRNA ligands that were expressed on the Najat nodules, so within those non-myelinating Schwann cells and the satellite glial cells, that could be being released from those cells and then go on to interact with the axons of the nearby sensory neurons that sprout into that space. So those would be receptors on those sensory axons. And so some of them that came back were things like osteopontin and clusterin, which have already kind of been implicated in axonal sprouting already in the literature. So we thought this was really maybe some targets to go after that might be really interesting for this sprouting mechanism and this idea of chemotaxis potentially happening with these sensory neurons. So we think that maybe these ligand receptor interactions that are going on between the cells at Najat nodules and these sensory neurons may be potential mechanistic drivers of pain. If these are actually neuromas, do they have this ability to spontaneously fire, which is similar to other types of neuromas? Do they have this ectopic activity, and are they signaling? Are they actually functioning? So we're hoping this will establish some grounds for future researchers to investigate. And with that, I'm just going to say thank you to all of the members of our group. We have a huge pain group here. I think between all the postdocs, PhD students, and undergrads, it's like 50 to 60 people. So we are a really great collaborative group. And yeah, thanks.
Thank you so much, Dr. Shiers, and to both speakers. What amazing presentations. We'd love to get some questions now.